Okay, the point of this little presentation, I think this is going to be one that you want to go back and look at maybe multiple times, just the PowerPoint of it if you're able to open that. And you should be able to open it in Google Docs, even if you don't have PowerPoint or OpenOffice on your computer. Yes. So I want to show you some of the features from the same batch of images that you're going to be working with, so that you can see little examples of what it is that these viruses and features look like. And just as a way to give you a little bit extra help. Okay, so we're going to be dealing with a coronavirus. The coronavirus is called MHV, which stands for mouse hepatitis virus, but every coronavirus essentially looks the same in an electron microscope picture. There's really no major differences there. And so here we have 5 images from cells, and they are absolutely full of little viruses. So to be a little virus, or to be something we can call a little coronavirus, look, you're generally going to be about this big, and it's generally going to look like a donut with a little white hole in the center. Occasionally you won't be able to see the white hole, but each of these things, if it's a real virus, is going to be inside some kind of single membrane vesicle. So there's going to be a little wall, like a little chamber outside of it. So something like this kind of looks like one, but I don't see that chamber outside of it. So it's actually not a virus. Our virus is going to actually form up in an organelle. So look at this and see if you can tell me which organelle our virus likes to use to assemble itself. Can you see it? It's the Golgi apparatus, right? And here's another Golgi. You'll have a couple little stacks up here. Here's a lovely Golgi with little virus inside some of the parts. And here's a little chamber from the Golgi just full of brand new virus particles. And there's some little viruses inside these little chambers. So some of these you can actually see the virus in the process of being made. They'll actually sort of line up at the edge and get pushed into the Golgi. So here's a nice one. Here you got one that's about halfway through being made. So all this stuff is just going to wrap up, and it's going to pop out into this little chamber of the Golgi apparatus. Here you can see the Golgi stripes. And that's going to be a new virus, just like this guy, or just like these guys, or just like that guy down there. Yeah, so that's how you recognize viruses. They're like little donuts. Now let's see, a virus is going to be about 100 nanometers. And so twice as big as a virus, to 3 times as big as a virus, 200 to 300 nanometers, are going to be the size of the little factory chambers where the virus copies itself. So we can call these the replication factory. And so you can count individual chambers. You'll see each of the chambers has 2 membranes on the outside of it. And each one of these is filled in with a little bit of, it looks kind of like cobwebs on the inside. You'll see they look quite different from mitochondria. So here's a mitochondrion, and see how dark that is? The actual virus replication factory double membrane vesicles are what we call them, but you can just call them the factory vesicles. They're going to have a double membrane, and they're going to look very light on the inside compared to mitochondria. Like down here, you can see mitochondrion next to a bunch of these little factory vesicles. Now the other neat thing is that sometimes if you see the factory, you will see the central feature. Oh, I almost dropped my microphone. You'll see the central feature in that factory, which is right here. It's called a convoluted membrane. So there's one here, there's one here, there's a little bit up here, a little bit over here, a little bit over there. And generally, if you can find one of those, there will be a ring of double membrane vesicles, like the factory, arranged around it. So it's kind of like grapes. I think of this as the grape stem, and these are the little grapefruits on the outside. All right, let's just hold the microphone. Okay, so in MHV-treated MHV-infected cells, you get some crazy things that are probably lysosomes. Let's look at some more of these. And so it's going to be a chamber, but with a bunch of stuff inside. Here you can see a mitochondrion for comparison. Look at there, another chamber with a bunch of just random stuff inside. Little, big, dark, light. And there's a mitochondrion, very healthy, nice-looking mitochondrion over there. Sometimes it'll just look like a bunch of twisted up membranes, or who knows what these are. Sometimes it's just like a little bit of everything inside of there. This is what a something like a lysosome would look like, roughly. Okay, so here we've got very nice looking, actually, rough ER and very nice looking mitochondria. Look, they're wearing their little horizontal stripes. There they are. Boom, 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 boom. And yeah, as long as these stripes are made of little lines that are pretty close together, then that's a pretty healthy mitochondrion and a good look. As far as the rough ER, look, you can see the individual ribosomes here, and you can see all of these little dots out here. Each of those is a ribosome. So you can see the cytoplasm's full of ribosomes. But some of the ribosomes are going to be attached to membranes, and these are just ribosomes that are going to be making membrane proteins for the most part. Now you can see one other feature in here. Well, a couple other features. You can see a nucleus right down there. Look at its nice double membrane. One membrane here, and the other membrane is kind of wavy on the outside. Everywhere that it pinches very close together, that's actually going to be a nuclear pore. That's where you would find one. And I see this guy. You recognize that? That's our lipid droplet, right? Yeah, you kind of, it's kind of like a word search or something like that. You almost got to let your eyes relax for a second, and you say, oh, what's that big blank spot? It's a lipid droplet. Kind of medium gray, a little darker than the background, and just nothing going on inside of it. Okay, let's look at Golgi apparatuses. So they don't always look quite as nice as in the images you'll see in a textbook. So these are Golgi apparatuses. They're going to tend to be on the thick side of the cell, which both of these are. Here you can see some of them that are stacked up a little bit. This is probably also Golgi apparatus. We're just catching it from a different angle. Because remember, they look like a stack of pancakes. And so if you happen to look at your stack of pancakes, instead of like cutting it in half, if you look at it from the top, then it's going to look like little round pancakes. Or if you look at it from kind of an angle, it'll seem like pancakes that are kind of stacked on top of each other, but you know, not perfectly. Here's another Golgi up here. So we got 1, probably 2, possibly 3, 4, maybe 5 Golgi apparatuses all in a pile there. And look at the mitochondria on here. Pay attention to how beautiful they look. And you can even see that thin little double membrane and the dark sort of staining inside. You can see the little individual ribosomes down here. And here's a little bit of rough ER with the little ribosomes on the edge. So yeah, looking great. Nice big mitochondrion up here. And there's the edge of the nucleus. And you can kind of tell it's the nucleus because it's got all these dark bits pushed up to the edge. That's all the messenger RNAs waiting to get out. And here's another nucleus, and you can see all the little bits pushed up to the edge, waiting to get out, and this might be a piece of a nucleolus here. Look at that. That's probably going to be a lysosome, and that's just, yeah, a vesicle with a whole bunch of really random-looking stuff going on the inside of it. But these are Golgis. This is how you recognize Golgis. Okay, so can you see the, this is an example of a really bad looking cell. I would try and find you something better. But you can still see, even though we're farther away, we're kind of zoomed out, you can see the little rough ERs, these little snaky tracts with the dark dots along the edge. They're pretty recognizable once you get to spotting them. The mitochondria in this image are not quite as dark relative to the background as they usually are, but you can spot them by their size and by their appearance. So there. Yeah, little mitochondria. And this zoomed in picture over here, you can see very nicely a bit of rough ER there. Looking lovely. And some free ribosomes up here. And this blobby gray thing, this may be a weird lipid droplet. But they're usually quite round, and so I don't really get what's going on with this thing. Yeah, I would say you could ignore that one. Okay, when you're looking at C75 treated cells, you're going to see some strange stuff. I'm going to show you how to identify it. So what you've got here are mitochondria that are having the worst day of their little lives. Yeah, look at this. Does that look like a healthy mitochondrion? You can still see the double membrane up here around the edge. And you can see that each of these little things would have been a stripe, but it's just grossly bloated. A normal mitochondrion size would be about this big. This is a fairly healthy one. And you'll see them in the same image and just say, whoa, that's weird. Look at this mitochondrion. It's actually got a giant hole in the center. You can still see the stripes. You can still see the double membrane and the darkened overall appearance. But man, does that look weird. And then in addition to the mitochondria over here, there's some very big, open, kind of shattered ones. And look, double membrane around the edge, so it's probably mitochondrion. These are little tiny condensed mitochondria. So this is another way that mitochondria can look sick. They can just be real small and like you squeezed them out, like they were a sponge and you wrung it dry. You see these guys? Boop, 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 boop. Those are going to be lipid droplets. And yeah, they just look like lipid droplets. They just didn't stain completely evenly in this case. But those are definitely lipid droplets. And you can actually see there's probably a lipid droplet over here where the center didn't stain very well. I think all the ones in the images I'm going to show you for the actual thing, I think they're all pretty evenly stained, if I remember right. Okay, some more, extreme mitochondrial weirdness, just like it says here, mitochondria with holes in the center, with actual little ribosomes poking through. There's another one with a hole in it, another one with a hole in it. This is a great big pork chop looking mitochondrion next to a little small condensed thing that probably is or was mitochondrion. And there's a couple of fairly healthy looking, although they're maybe a little bit squeezed out, mitochondria here. When you're looking at a mitochondrion and trying to decide if it's healthy or not, I would say you just go with your gut feeling. Look at something like this. These are pretty obviously not okay. Like a really weird shape, then it's something's wrong with it. And a weird appearance like on the inside, if you see little circles or little ovals instead of little stripes. So these stripes actually look pretty good. This part of the mitochondrion is healthy, but since it has this giant hole in the top of it, we know that it's not completely healthy, and I would mark that down as sick. Yeah, I think most of these on these three images I would mark down as unhealthy. There's a neat thing that happens to the Golgi apparatus in C75 treated cells, and so I want you to see if you can spot it. What happens is, I call it the SpaghettiOs phenotype. The Golgi just shatters into a bunch of little tiny droplets. They still hang out where the Golgi used to be, but look at them all. There's another little patch of them, and there's another little patch of them. They'll still be on the fat side of the cell, and after you've looked at the healthy cells, which are at the beginning of your assignment, then go and compare those to the C75. So if you see something like this, I would call it a Golgi that is aberrant, that is not healthy. And if you see the regular ones with stripes, they look quite different, and those would be normal, healthy Golgis. All right, and just so you can see it. So here's some kind of a mix of fairly normal-looking mitochondria with normal stripes inside of them. This one is gigantically large, which probably means it's on the way to being sick, but I can't see anything specifically inside of it that looks super weird, so I guess we have to say it's, reluctantly okay. But look at these little lines up here, just everywhere. That is tubulin. This cell has gone on a tubulin craze and is just massively overproducing cytoskeleton more than it has any use for. And so this is another one of the things that I think C75 does more often than not. Won't do it in every cell, and every cell is going to look a little bit different, but overall, it should do it statistically more often than in untreated cells. And statistically is the only way we can make any kind of actual judgment in science. Do you see the rough ER here with little ribosomes all along the edge? Looking great. And some free ribosomes just floating around here. Even a couple of free ribosomes here in this forest of cytoskeleton. Very strange. You got the edge of the nucleus right up here. Okay, so that's your little help file, and I would say keep that on hand so that you can flick back to that as you are looking at each of the cells in the mini project file, which is also included in this part of the Blackboard site. And yeah, use those, have a look, go to tutoring, talk to Caleb about it. If you've already done that, you still need more help, come see, make a little Zoom appointment with me. But watch these videos and try and see what you can do to figure it out. I can't tell you a mitochondrion looks exactly like this. Just the same as I can't tell you a person has to look, exactly this tall and exactly this shape. There's some variation, but humans are very good at categorizing things even in spite of variation. And so that's your task here, just to do your best, your level best on this. That's all I'm looking for. And then once you have your data, you're going to do some statistical tests on it to see whether or not you can support the hypothesis that C75 causes some changes, or that virus infection causes some changes inside the cell. But you got to support it with stats, because that's the only way to support anything in science. So there we go. All right, thanks very much. This is the end of this section.